In this set of lectures, we're going to discuss some of the molecular properties of the cells that are responsible for sensory transduction. Today, we are going to discuss some specialized neurons that are able to transduce light signals to electrical signals. These are the photoreceptors. In the animal kingdom, we find many types of photoreceptors. Some are very simple and some are very complex. But at the molecular level, all these photoreceptors use the same kind of molecular mechanism to be able to detect light signals and somehow translate these light signals into some form of electrical changes. Here are some few questions about phototransductions. For example, how rods are built for maximum light sensitivity? Can we really see single photons of light? What are the steps in the phototransduction cascade where there is amplification of signal? What determines the termination of the phototransduction cascade? How fast these photoreceptors can respond to light signals? What's the energy cost of the phototransduction cascade? What are the main types of ion channels and transporters in these photoreceptors? How photoreceptors adapt to light? What's the role of retinal epithelial cells in light adaptation? What are the differences between rods and cones? Are cones really color sensitive? Why we cannot see well objects that are blue? How photoreceptors release neurotransmitters even without fire action potentials? Finally, why some blind patients still can respond to light? We're going to answer some of these questions in our lecture, but if you have other questions, please bring these questions to our class. The stimulus for vision is this very narrow band of electromagnetic radiation we call visible light. It spans from about 390 nanometers, which corresponds to violet, all the way to 760 nanometers, which corresponds to red. We are not able to detect very well using photoreceptors, infrared radiation, or UV light, although some animals can detect these other types of electromagnetic radiation. The major types of retinal cells that you find in the retina are shown here in the simplified diagram. And you can see in the top of this figure that you have rods and cones, which are the photoreceptors, that are closely associated with this layer of epithelium that we call pigment epithelium. Then these photoreceptors synapse to other types of cells, which correspond to bipolar cells and horizontal cells, and then these bipolar cells synapse to the ganglion cells that send the axons to form the optic nerve. So the electrical signals travel from photoreceptors to bipolar cells to the ganglion cells, and then the ganglion cells send the signals via the optic nerve to the brain, where these visual signals is going to be processed and interpreted as light signals. Now, there are two major types of photoreceptors. There is rods, and then there is cones. The rods have this more rod type of structure, and the rods are there's many more rods than cones in the human retina. There's about 100 million rods in the human retina and about less than 10 million cones in the human retina. Both rods and cones have this domain that we call outer segment, where you find this membrane specialization that we call discs. And you can see in the rods that the discs are separated from the cytoplasmic membrane, while in the cones, those discs are contiguous to the plasma membrane. Then there's a connecting cilium that connects the outer segment to the rest of the cell, and you can see that the rest of the cell encompasses the inner segment and the synaptic ending. The inner segment have the organelles such as mitochondria, while synaptic ending have synaptic vesicles, and the photoreceptors uses glutamate as neurotransmitter. Traditionally, we divide the functions of rods and cones as rods mediating what we call monochromatic type of vision, while the cones mediate what we call chromatic vision. Rods have only one type of visual pigment, while the cones have in the human retina there is three types of visual pigment. This type of monochromatic type of vision is called scotopic vision, and they can operate at very low light levels. While the phototopic type vision mediated by cones usually involves higher levels of light. And the rods are involved in this night type of vision, peripheral type of vision, while the cones are mediate what we call everyday type of vision that involves detection of details of objects, color, and motion. We know a fair amount about the photopigment that's found in rods. This photopigment is called rhodopsin, and as you can see in this figure, the rhodopsin is actually chromophore, in this case 11-cis retinal, that's covalently bound to this lysine residue in this opsin that has the generic name as rhodopsin. So and as you can see, rhodopsin, the protein part has the structure that's very similar to a G protein coupled receptor. It actually was the first G protein coupled receptor to be cloned, and we know in very good detail how this protein, the structure of this protein. The only part light sensitive structure in rhodopsin is the chromophore, in this case 11-cis retinal. As you can see by the structure of the 11-cis retinal, when it's bound to rhodopsin, this molecule is composed of several carbon atoms with single and double bonds. And when light strikes 11-cis retinal, it can isomerize this 11-cis retinal to this another conformation that we call all-trans retinal. And this reaction is very, very fast. The isomerization takes few hundreds of femtoseconds. And if you look at the figure on the right, there is a plot of the absorption in the y-axis, that's called extinction coefficient in this case, plot against the wavelength in the x-axis. What you can see that the 11-cis retinal absorbs mostly wavelengths about 360 nanometers, which is UV light. But when the 11-cis retinal is bound to rhodopsin, now this 11-cis retinal is able to absorb actually visible light about 500 nanometers. So 11-cis retinal by itself is not sensitive to visible light, but the 11-cis retinal, when it's bound to rhodopsin, the amino acids that surround this 11-cis retinal tunes this 11-cis retinal to become sensitive to visible light, about 500 nanometers. And the 11th retinal, when is activated by light and is bound to rhodopsin, then it will initiate a cascade of events in the photoreceptors, and this cascade of events we call phototransduction. There are several points that I would like to point out. First is that oddly enough, in the dark, these cells, the rods, are relatively depolarized. So if you measure the intracellular membrane potential of these cells sit about minus 40 millivolts. Uh, but when this photoreceptor is exposed to light, these cells hyperpolarize to about and depends on how much light strikes these photoreceptors, but they can hyperpolarize to levels about minus 60 millivolts or so. And finally, what has been learned using various biochemical assays is that psycho-GMP is the intracellular cycle messenger for this phototransduction. And how this phototransduction happens is shown in this figure on the right side. You can see that in one, the light stimulation of rhodopsin leads to activation of a G protein, in this case called transducin. This transducin G protein, when it's activated by rhodopsin, will break down in alpha GTP and beta gamma. The alpha GTP then can bind to phosphodiesterase, which is an enzyme that is also found in those discs, and this phosphodiesterase will break down GMP, psycho-GMP to GMP. So activation of rhodopsin ultimately leads to a decrease of psycho-GMP levels. And psycho-GMP is important because it is able to bind to psycho-GMP gated channels. As you remember for our lectures of ion channels, psycho-GMP channels are channels that are selective for both sodium and potassium. So when these channels are open in the dark, you allow the sodium influx, and that's why the cells are depolarized. When the photoreceptor is exposed to light, then when the psycho-GMP levels decrease inside of the cells, the psycho-GMP gated channels close, and therefore these cells hyperpolarize. This current via psycho-GMP gated channels, so the sodium and influx via psycho-GMP gated channels in the dark, equal current leaves the cell via potassium channels, and this loop of current that you see in rods and also in cones is called the dark current. Because there's this continuous influx of sodium and efflux of potassium, there is a large energy cost for these cells to keep this condition, and the energy cost is mainly because you have to give ATP to the sodium potassium ATPase to keep this steady state ionic conditions in these cells in the dark. This influx of sodium and efflux of potassium under dark conditions is called the dark current. So here is one exercise. You can see there is a figure plots voltage in the y-axis and time in milliseconds in the x-axis, and supposedly you're recording from a single photoreceptor in current clamp mode. The record is done in the dark with a brief 1 millisecond flash of light at varying intensity. Draw the expected trace with time in the x-axis and the voltage at the y-axis. Likewise, repeat the same experiment now in voltage clamp mode, holding at minus 40 millivolts. Draw the expected traces with time in the x-axis and current in the y-axis.